So you're more into mathematics and science, okay? Is that right? Good. So I will be very simple at the beginning so that you progressively move in. But I'm not good in engineering. I cannot open this thing. Ah, here. So you don't have to be ashamed not to be good in biology. Good. What I will try to do with you, I think I will have about 5 or 6 lectures with your group, before the summer. What I will try to do with you is, ultimately my goal is to make you understand how the vascular system functions. But before we do that, we have to think a little bit more in simple terms and wonder, okay, this is a class in physiology. What is physiology about? Okay, that's what I will try to get today. And it all boils down to the problem of the complexity of our body. If you take a very simple, unicellular, one cell, we'll see later what a cell is, a very simple form of life in water, in the sea, for example, plankton, whatever. Those cells, and we will talk about what they contain in a moment, but those cells have a relatively easy life because they will pick up foodstuff from the water, they will transform it, metabolize it to make their own material, and the breakdown products, the garbage, they will throw in the water. To our life, the cell will go to the supermarket, buy things to cook, and then throw away the garbage. Very simple life. No more foodstuffs here, too much garbage there, the cell moves, goes somewhere else. But we cannot do that, because we are a very complex, multi-cellular. We have millions, billions of cells in our body, and they cannot move around. So to be able for each cell to go to the supermarket and have a garbage can, we have to construct a complex system. And the knowledge of that is physiology. So what is physiology? Has any one of you learned Greek, ancient Greek? I would presume no. Maybe some Latin, I'm not even sure. But in Greek, physis means to be, to exist. And logos is knowledge, science. So physiology is the knowledge of existing physically, not in the brain, not the philosophy, not the soul, just physically. It's the knowledge of the function of the body. And from day one in this WCU program, I have argued with my distinguished Korean colleagues that functional physiology is not a good name, because physiology means the knowledge of the function. Okay? So, but I've lost that battle, and I've lost many battles in my life, so it's not crucial. So physiology is the science, and you have access to those slides, don't you? So, okay. The science that tries to understand how life is possible looking at a cell physiology, or at more complex, as the tissues, we'll see that in a second, the organs become more complicated. And thus physiologists try to understand how the body functions under normal conditions, normal physiology. And this is what we talk about in this course. But obviously, physiologists also try to understand what can go wrong when we become ill, when disease strikes. We all know if you have a fever, your body doesn't work very well. You know? If you've had too much to drink the night before, your body doesn't work very well. You know? So to try to understand how the body functions under abnormal conditions is also physiology. But we call it pathophysiology because pathos means suffering, disease. You know? So this is what physiology is all about. And what do physiologists like myself, although I'm more considered to be a pharmacologist, you'll understand why later, what do physiologists like myself do? Well, we look at how cells behave, how individual cells interact, how organs function, how different organs allow the body to run around, to live, to procreate, etc. Now, this implies a very profound knowledge of a number of other sciences, among which anatomy is probably a very, if not the most important one. Remember, by definition, physiology is a translational discipline. You know, to understand how the body functions in health and disease, you have to know from the cell all the way to the integrated system. And this is something that young people often ignore, because those of you, very few of you, are involved in biological research, but you look at your little cell, you look at your cell culture, and you look at the tree, you don't look at the forest. You know? And it is the forest that lives. It's not the single tree or the single leaf of the tree. Okay? You understand that? Oh, I forgot to say 3 very important things before starting. One is, I don't want you to sleep in my class. So if you're very tired and you want to take a nap, don't come to my class, because I get very angry if I see you sleeping. The other thing is, I don't want you to play with phones or computers. You have to listen. Because everything you want, you should know, will be on those slides that you can access later. But I want to look at you and have the illusion that you understand what I'm talking about. The third, most important, so no phones, no computers. Again, I get very angry if I see you play with the phone. You know. My phone is silenced. I have one too, but it's silent. Are you very tired, sir? Are you very tired? If you're too tired, you can go outside, okay? It's very rude. Do you know it is very rude, what you just did? You have a lecturer here that comes from far away, gives a lecture, and you're yawning as if life was so terribly difficult. This is not very pleasant for the lecturer. You have to learn that. The third most important point is, you have to ask questions if you do not understand. If you do not ask questions, I can assume that you understand everything that I say. And don't forget, there are no stupid questions. There are only stupid answers from the lecturer. Okay? Don't forget that. The Chinese say that if you ask a question, you may sound silly for a few minutes. You may sound stupid for a few minutes. If you don't ask questions, you will be stupid all your life. So please, if you don't understand, interrupt me. Okay, so now we're back on track. So, as I just said, modern physiology cannot exist without knowledge of other disciplines. Anatomy is the first one, because historically this is how science started. Anatomy, well, it comes from another Greek word, anatemnein, which means to cut. And the knowledge of the structure of our body came from people who, in the Middle Ages and later, or before, even started to cut dead bodies to see how the structure was. And I'm always very proud to show this slide, because the first and probably the best anatomist of all time, Andreas Vesalius, came from my home country. So we are very proud of him in Belgium. And he set the tone, and he defined, proper anatomy. And his book, that of which this is an illustration, is one of the most wonderful books, medical books that you've ever seen, published in the Renaissance. Now, besides, why doesn't that work? Does that work? Ah, I see. What do? I go to the previous one. Besides anatomy, the physiologist also will use approaches of pharmacology, but we cannot dissociate physiology and pharmacology, we'll see that later, of chemistry, of molecular biology, and nowadays of genetics. Again, it's a translational science, so we need to know it all to understand how the body functions. Good. What's a body made of? Well, if I ask you what's a house made of, I think in Korea you use bricks, don't you, to build houses. Well, it's the same in our body. Our body is made up, well, first of all, it's almost all water, it's 90% and more of water, but we have bricks, the cells, and fluid. And the basic unit, the cells, are all derived from the one cell. We have to keep that in mind. Although we have so many different cells in our body, genetically, in terms of genome, they all come from the one cell, the fertilized oocyte of your mother. That's what it all started. That start multiplying and differentiate, and that's beyond the scope of this course to go into that, but that's one of the most fascinating things, of course, how this one cell start to differentiate and eventually will form nerves and muscle and all of that. So we have cells, billions of cells. We have juice, fluids, plasma in our blood vessels, in cavities around the heart, in cavities around the lungs, in our joints, we have fluid. And the cells can float in the fluid. That's blood. Blood is fluid, plasma, with cells floating in it. But under most circumstances, or in most organs, the cells adhere to each other. They clump to each other. And they form what we call tissues. And different tissues, and those tissues can be soft, quite soft, or hard, the bone, cartilage. And those tissues form specialized structures, for example, blood vessels. And then different tissues will group to form an organ, the heart, the brain, the kidney, the gut, the stomach, etc. Okay? That's an example of a cell, you'll see that picture later again, of a cell floating in fluid, blood, a red blood cell. Blood contains red blood cells, white blood cells, and platelets. Different cell types. Oh, about 10 different cell types, but they all float in the blood. This is an example, and again you will see that picture over and over again because it's one of my favorite pictures. This is an example of a tissue, a blood vessel. Here you have fat. Here you have muscle cells. Those are nerve cells. This is connective tissues. So different cell types combine to form a tissue. And this would be a schematic view of that, of cut through a blood vessel wall with one cell type here, the endothelium. And we'll talk a lot about the endothelium because that's what I spent the last 30 years of my life looking at. We have connective tissue, we have smooth muscle cells, we have nerve cells, we have again connective tissues, here with other cells, and here there would be fat. And when we put those different tissues together, then we have one organ or organs. And for example, the blood vessels put together form the vascular system, departing and returning to the heart. But we will see that in later lectures. Okay? So you start from one cell, you make tissues, and you make organs, and you make a system. This is the cardiovascular system. So what do the physiologists do? Well, they try, as I said, to determine the normal function. And they can do that. They can do that for a cell, as I said, cellular physiology, a tissue, an organ, or an organism. For example, in the lab on the 4th floor, we have people that do all of that, because some of us look at cell cultures, some of us look at isolated blood vessels, some of us look at a more complex system. If you want to know more about that, you're always welcome to visit with your colleagues on the 4th floor of the other building. Okay? Good. So we try to understand how that functions. And how do we do that? Well, there are basically 4 approaches. One is we look at the cell, the tissue, the organs, the system, and we measure its activity. We try to figure out what it does. That's one way. The other way is to remove it. If you have a cell type, an organ, and you remove it from the body, and you see what goes wrong, because we need every bit and piece that we have to function, you can understand the function of that particular cell type or cell group. And I will show you examples of that. We can, and that's, I said already, that it's very difficult to dissociate between physiology and pharmacology, because physiologists use drugs all the time, and pharmacologists have to understand physiology. So we can use pharmacological interference. Again, you'll understand later what this is. And then more recently, the last 20 years, we have learned to manipulate genetic information. So we can modify the genome of animals. And this is a way, of course, to understand better how the body functions. And I will today try to give you a few examples of all of that. So, measuring activity. What is cellular activity? Well, if you want to understand what the body does or how the body works and why we can stand up, run around, survive, fight, learn, and all of that, we have to understand we have to know the major cellular activities. And those are all exerted, those activities, by specialized cells that have evolved from this one oocyte that have differentiated to do this specific thing. One of those basic functions is contraction. Skeletal muscle, we will see the heart, or in the blood vessel wall, or in the gastrointestinal system, in the bladder, in the lungs, where we have a number of cells that can contract and relax. And that will help determine the activity. Secretion. Those are cells that form a substance and then secrete it in the blood or in the outside world. In the blood, we call that endocrine glands. And we will see examples of that. In the outside world, we call it exocrine glands. We will not talk a lot about that, but exocrine glands, for example, are sweat glands, milk, breast glands, A number of those are exocrine glands. In our gastrointestinal system, we produce a lot of enzymes to digest. Those are exocrine glands. Because, as I will say probably later, if you look at the body, the inside of our gut for the body is the supermarket, is the outside world. So if we want to have carts to get the food, we need enzymes to prepare the food to be able to take it up. You understand? So, exocrine gland. Conduction. If my brain decides that I have to lift my foot, how does my foot know that? It's because there are specialized cells that conduct electricity-like activity, and those we call neurons. There are cells that are energy machines for making, that are chemical machines to take up or to take the substances that we get from the supermarket and transform it in our own substances. That's the liver. Or store it for a rainy day. That's fat. So those are metabolism and storage, very important. As I said, we have to get rid of the garbage. And there are cells that specialize in doing that. And the most typical example is the kidney. The function of the kidney is to get rid of the garbage. But it's not only the kidney, it's also the liver in part with the bile formation, or the gastrointestinal system. When we go to the toilet, we get rid of garbage. So that's how we function. And then a very important function too is not only is exchanges, and I only list exchanges here, but I should really be more broad and say exchanges in general, gas in the lungs, and in the gastrointestinal system, uptake of foodstuffs. There are other functions. For example, if you cut yourself, it will heal. This is physiology too, of course. And if we grow, we form new blood vessels. There are a number of other functions that I cannot go into today. But for the understanding of what I will talk about in those lectures, this is the list of things that you have to remember. Okay, what's a cell? Well, a cell is basically a drop of living substance that we call protoplasm, and that is surrounded by a cell membrane. And if you look at it under the light microscope, as one was able to do when I was your age, electron microscope was just beginning at that time. Hard to imagine, but you saw the cell membrane, then you saw what we call the cytoplasm or cytosol, and that's a blurry substance, the things floating in there, and then there was in the center the nucleus with another cell membrane, and there was a very marked point there that we call nucleolus. Okay, again, this is how it looks. You can go to any microscope and take any cell and you will see that. Now, as the techniques and electron microscopy progressed, things became a little bit more complex. And we now know that besides the cell membrane surrounding the cells, there are membranes surrounding the nucleus, there are membranes surrounding the mitochondria, which is here. So there are several membranes inside the cells. And in that blurry cytoplasm, those cell membranes delineate what we call organelles, smaller subfractions, but constructed. There are specific functions and specific structures inside the cells. And the one that I don't think we will talk about it, but that you will hear most about it are the Golgi apparatus, lysosomes, mitochondria, the ribosomes, and there are a number of things, eventually microfilaments and endoplasmic reticulum, sarcoplasmic reticulum. I mean, there are a number of those. All of those we discovered in the last 50 or 60 years, have very specific function and are very important. Now, each one of those organelles could be the topic of 5 or 10 lectures, but I cannot do that. But again, remember, it's very complex, and once in a while we'll come back to some of those substructures. If we want to measure activity of a cell or of a tissue or of an organ, we have to know how can one change, or does in our body the activity of a cell change? Well, in the rule, it is because the cell receives or perceives a signal to change its level of activity. If the light goes out, I go to sleep. That kind of things. Now, how do I go to sleep? That involves a chain reaction of events. But it's basically like that. If I see a tiger, I have two options. Lay down or run. That's the signal. Then a chain of events follows that will lead us to change the activity of the body. Okay? Good. So ultimately, a group of cells has to perceive a signal. Now, at what level in the signal, in the cell, can we change the activity of the cell? And this can happen at the level of the cell membrane, and we will see that there are basically three major possibilities there. Or it can be because we get inside the cells, and again there's several possibilities. Let's look at those. The cell membrane. The cell membrane is a wonderful thing because it encloses the cell and separates the inside of the cell from its outside world. And the membranes of our cell types are different because they have adapted to their specific function. Now, how can they do that? Well, they can adapt because, well, that cell membrane is basically a layer or bilayer of lipids. Lipids here, lipids there. And this would be three-dimensional there. But in those lipids, anchored are proteins. And those proteins can be mainly totally inside the cell membrane, or can pop inside the cell, or through terminals can pop outside the cells. And the thing is that pop outside the cells can capture things and then can transfer the information inside the cells. That's the basic principle. Now, what are the proteins? And those are always proteins that perceive signals and pass them on. What are the proteins? Well, they're basically in the outside system, in the cell membranes, there are 2 or 3 types of function. How shall I explain that best? The first subgroup, let's go back 1 or 2, sorry. The first subgroup would be receptors and ion channels. The third subgroup would be transporters. We'll talk about intracellular receptor signals as well. So, at the cell membrane, we have 2 types of proteins that can react to outside signals. And those will either be what we call a channel protein that will assemble to form holes so that something can get inside the cell and then close again. Or will be receptor proteins that will note the signal here and then transduct the signal to the inside of the cell. Now, those proteins can be coupled to G proteins or directly to enzymes, but we won't go too much into that detail. By the way, each membrane inside the cell also has such proteins that can, react to signals. But there, in this case, in case of nuclear receptors, the signals should be in the cytosol, not in the outside of the cell. Understood? You sure? No questions, so you understand. That's our deal. Eh? So don't blame me if you get very difficult questions at the test, okay? Because you didn't ask questions, so you understand it all. Not my responsibility. Okay, let's look at the different possibilities. What we call receptors are proteins that will recognize a substance. And that substance we call an agonist. This is pharmacology, by the way. This is not physiology. This is pharmacology, but we cannot separate it. So, a substance in our body, for example, a hormone, we'll see later what a hormone is, will bind to the receptor, and this then will change the function of the cell. And we will see in a second how that is possible. Now, pharmacologically, what we can do is we can design chemicals that can make this interaction impossible. That's what we call an antagonist. We also have ion channels. Those are holes that permit the flow of ions from the outside to the inside, mainly calcium, or from the inside to the outside, mainly potassium. Those are the 2 big players. And again, we can have substances that can make the channels bigger or block them. We also can get inside this. Oh, let me take the last one first. There also are proteins on the cell membrane that recognize something, capture it, and transport it inside the cell. Very important for certain foodstuffs and for certain important molecules. Those we call transporters. And then finally, as a pharmacologist, since ultimately all functions of the cell are based on enzymatic reactions. You know what an enzyme is? Who knows what an enzyme is? Come on in the back there. Who knows what an enzyme is? Yeah.
Enzymes of protein catalysis.
Yeah, those. You may not know biology, but you may know some chemistry. And if you know chemistry, you've heard of catalysts. You know, those are substances that accelerate a reaction. Well, enzymes are our own catalysts. They are proteins that catalyze, that accelerate reactions. We'll see an example of that. And so we have those enzymes, and there again, you have a substrate that is transformed into what your end product is, and you can interfere with that by blocking the enzyme. You can give it a false substrate. You know, you can design a substance for which it has a bigger affinity, but it will not form the product. And we don't need to talk about prodrug at this stage. So those are the different possibilities. And if you want to know a little bit more about those, there are on our every cell membrane of our body, there are tens, hundreds of such proteins that can capture signals. Now those are the most frequent receptors, and we will certainly talk about acetylcholine receptors, we will talk about adrenergic receptors, we will talk about histamine, we will talk about serotonin, we will talk about a number of those things. There also are a number of ion channels, and those are very important to understand changes in cell membrane potential. You know, as the cell membrane, I haven't said that, one of the characteristics of a cell membrane is that it is polarized electrically. And to keep that polarization, we need to establish an ionic equilibrium. There must be more positive or negative charge, one side or the other, if we want to change the cell membrane. Well, those ion channels, of course, play a key role in that. Who has a phone that is working there? Huh? Okay, good. So next time, before you come into class, you turn off the phone, okay? Good. What was I talking about while you were turning off your phone? What was I talking about? What was I talking about? Good. So despite the phone, you listen. Good. Okay, so I'm not totally mad at you. Good. Very good. So we can alter those exchanges and set the membrane potential. Again, we will see examples of that. Okay. Let us now begin to give you some functional data. This is a very simple experiment, like we do on the 4th floor almost every day, I think. We take a blood vessel, we cut it into rings, and you will see this slide many times. We cut it into rings, and we suspend it between a support and this system here, which permits to measure the degree of contraction of the muscle cells in the artery. The artery, we will see that, is round. It has muscle all around, and when the muscle contracts, the blood vessel comes straight. The tube becomes smaller, and you can measure that. And we do that. And then here, at this stage, we give a substance, Again, you will hear more about prostaglandins, prostaglandin F2 alpha, and we see that the preparation contracts. There are receptors on the cell membrane of the vascular smooth muscle that recognize the presence of prostaglandin, and immediately there is a change in activity of the tissue. Okay? Illustration of that. Now, here we start giving another one of those substances, acetylcholine. And you can see that the blood vessel relaxes. Another indication, a cell type in the blood vessel has recognized the presence of acetylcholine and has told the smooth muscle to relax. Now, the intriguing part of this experiment is that there are two rings of the same artery. But in one, we have removed the endothelium, and we don't see the response anymore. We'll talk about that later. But it illustrates something that I've said already, that is, one way to do physiology is to take the cells or the cell group away. Here you see a reaction, you take away the endothelium, you don't see the reaction. So, illustrating how with simple experiments you can get a better understanding. I already can tell you that this observation, fortunately not by me, but by Bob Fersgott in New York, gave him the Nobel Prize. A very simple experiment, but demonstrating how the presence of endothelium is absolutely crucial to see the response to acetylcholine. Okay? So here we demonstrate how a physiologist can measure activity, but to do so, he or she usually needs pharmacology to get the signal to the cells. This is another example where you see that acetylcholine causes a response. Again, we won't go into that right now. Good. Those were receptors, prostaglandins and acetylcholine, etc., and many others. What about the enzymes? Well, the enzymes, there are. This is not an exhaustive list. There are so many enzymes in our cytosol or in the different organelles that contribute to cellular activity. And every time we interfere with one of those enzymes, we alter cellular activity. And in disease, this becomes very important. Different activities of enzymes usually are at the base of pathological situations, of disease situations. The same for carriers. You see there are many carriers there, and those, again, it's not an exhaustive list. Every week we open Science or Nature, the big scientific journals, and we find other possible targets that can induce cellular activity. Okay, so the substance, this is the cell membrane, we have the receptors are activated. How do the receptors now conduct the signal inside the cell? Well, they can do that in different ways. In certain cases, they are directly connected to the second level of control. But in most cases, the proteins on the cell membrane are coupled to the intracellular target through other proteins that we call G proteins. G protein coupled receptors. Last year's Nobel Prize. So very, very important. And the receptor then will activate the G protein that will modulate the activities of enzymes inside the cells. And then those enzymes will generate the second messenger. Now, the second messenger, in the case of this enzyme, it would be cyclic AMP. Here it would be cyclic GMP. Where is cyclic GMP? No, here, cyclic GMP. This would be cyclic AMP or inositol phosphate. There are several possibilities. And then goes to the next step, which usually are enzymes that we call kinases. And these then eventually will lead to the activity change. So it's a very complex signaling cascade. And believe me, this is a very simplified form. If you don't believe me, open any magazine and you will see that it's a lot more complex. But as long as you understand that once the receptor is activated, then there are step 1, step 2, step 3, and down the chain, the cellular activity change. Now, the reason why you have to remember that is simply that this implies, again, if you have some knowledge of electronics or some knowledge of mechanics, you will immediately understand that the fact of having those different layers permits very good feedback control and, fine-tuning of the system. It's almost never an all or none response. It is at each level modulation is possible so that we can come to the proper response of the cells. What happens then? What are the consequences? Well, if you look at ligand-gated ion channels, those will increase the changes cell membrane potential and cause hyperpolarization or depolarization leading to cellular activity. This is very fast, in milliseconds. That's the fastest. If you change a channel permeability, boom. That's as close to all or none as you can get in physiology or pharmacology. Ions can also be governed not directly by the ions, but by receptor modulation, and this at the same time as G-protein coupled receptors leading to protein phosphorylation will occur in the course of seconds. The responses that I just showed you with acetylcholine, like this, for example. You see, we give the drug, and it takes a few seconds to get to the response for each concentration. That's an illustration of that. If we go inside the cell, where we start modulating the protein phosphorylation, and we go to gene transcription, we look at hours in terms of reaction. And finally, if we go and activate nuclear receptors and change the genomic information, then it takes even longer. So the reason why I showed this slide is to illustrate to you that changes in cellular activity can be very rapid, slow, very slow. For certain things, you have to wait an hour or two. For certain things, you have to wait 24 hours before you can see the effect. So there is a gradation. Again, different levels of regulation, where this would be for short-term, immediate, life-saving things. The lion is there, I have to start running. I cannot wait for 2 hours that my nuclear receptors are activated. I have to do something right now. And then progressively, this, for example, would be, I have a microbe in me, I have a cold, I have to generate resistance against, I have to protect the tissues, I need some inflammation there. That takes a little bit more time. So if you catch a cold, it takes several hours before the inflammation starts. See? difference. You know all of that, but it's based on very strict cellular regulation. And in all of those things, if we go back one, in all of those changes, ionic changes, calcium turns out to be, if not the most, well, I would say the most important ion. For some unknown reason to me, a reason unknown to me, calcium is the central activator of cellular activity. In all tissues that can be excited, when we say that can be excited, that is, that can react, calcium is always the key. An increase in calcium concentration leads to increased activity of the tissues. And I used and still used to call, when I write about it, calcium the activator ion. And so, good illustration of learning process. We will not talk about memory, but this is a good illustration. I had it read the first time. Now, how does calcium, we will see, we will understand that better later. Well, calcium, ultimately the cell does everything it can to keep the calcium low, so that the cell can rest. It's like you in the lecture room, You like to rest. You don't want to be too active. So you keep your calcium low. And activating a cell then implies that we will increase calcium. Now, where does the calcium come from? It can come from two sources. It can come from the outside, but we also, in the endoplasmic reticulum, or sarcoplasmic reticulum, we have a big storage of calcium. And thus, if the cell has to be active, it needs more calcium. And the signals will lead to activate the cells will either increase influx of calcium from the outside or release of calcium from the inside. Typically here through calcium channels, and here typically through second messengers, inositol phosphate in particular. And it's a little bit more complex than that, but this gives you the feeling. And then once the signal for activity is terminated, then the calcium has to be pumped out. And it can be pumped out in the outside or pumped in again in the sarcoplasmic reticulum. And thus, the signal, for example, acetylcholine, will come here, open and do all those things, and the cell will be active. The moment acetylcholine is removed, and it is removed because it is degraded, it is broken down, we'll see examples of that later, immediately the signal stops, and the cells start pumping out calcium back and pumping calcium into the sarcoplasmic reticulum. Do we understand that? Okay, so if I ask a question about that, you will be able to answer. Good. I'm pleased to know that. Back to my preparation here. In this instance, this shows you what happens if I take doesn't matter, forget about the endothelium for today. What we do here is we give this substance that we call a calcium ionophore. A calcium ionophore will augment the permeability of the cell membrane, and although there is no activation signal, allow calcium to get inside the cell. Okay? So it's an artificial way to force a calcium increase inside the cell. And sure enough, you see a very profound response, very profound change in activity. And again, we can measure that using specific techniques. This would be the very low calcium level in the absence of activation. This is with acetylcholine, this is with the calcium ionophore. And we'll come back to the rest of that slide later. Okay, now how can we, thinking about the vasculature, start playing with that? Well, we're back into pharmacology, and we're back at drugs or hormones that activate receptors that will lead to depolarization and calcium increase and thus contraction, or we will have substances, hormones, that act to block the channels or block the calcium channels or open the potassium channels, other receptors, and again we'll see those, and those then, through those coupling, will inhibit contraction and lead to relaxation. Every smooth muscle cell in our body is continuously in balance between vasoconstrictor and vasodilator signals. And it is the cell itself that integrates those different signals and leads to the proper level of activity. Now, to do that, to be able to be active, to have the contractile proteins that interact, to secrete the hormone, to store the fat, we need gasoline. We need energy. And the energy in our body comes from the production by those little beasts, the mitochondria, of ATP. ATP is the key. Whenever it splits off a phosphor, and phosphorylates another enzyme, energy is made available, and we can move on. Okay? Again, I will not give you a course in biochemistry. I will probably not be capable of doing it, because I can tell you when I was in medical school, biochemistry was very simple compared to nowadays. But this is still, 50 years later, this is still true. This is our gasoline, ATP. And ATP is formed by the mitochondria and then can be used for the different things. If you're interested, you can look at that slide. I will not ask you a question about details about this, I can tell you, just to be complete. And ATP just can go to the endoplasmic reticulum and store calcium or produce other substances. It can synthesize proteins. It can provide energy for muscle contraction. It can provide energy for transport. It is the key. If we run out of ATP, we run out of gasoline, and we die. It's that simple. Okay. So, a physiologist can measure activity at different levels, and it can be very complex. A physiologist can also remove it from the body. Claude Bernard, one of the founders of modern physiology, he was French. He did his work around the middle of the 19th century, and he has said in French, but I will show you to you in English, in my mind, the most important statements in physiology. You only know what a nerve or a cell or a gland or whatever does after you have cut it. If you don't cut the nerve, you don't know what the nerve does. And you know that if you cut a nerve, there are very hard consequences. But that's one way to know what the system does. And actually, I've already shown you that illustration. Remember, here you see this wonderful relaxation. We remove the endothelium, we cut the nerve, response is gone. That's a very typical example of Claude Bernard's principle. We can also use pharmacological interference. Now, this is a very complex and sophisticated way to say we can use drugs, selective and specific drugs. What is pharmacology? Well, we're back to ancient Greek. Pharmakon. When you talk when we talk about pharmacy, we think about medicament, we think about drugs to cure disease, don't we? Well, pharmakon, the original word behind pharmacy in Greek, doesn't mean therapeutic agent, it means poison. So pharmacology emerged from the knowledge of poison. To give enough poison to kill your enemy, or enough to become insensitive yourself, you see, which brought in the concept of dose response. And of course, pharmakon, logos, so pharmacology, in Greek means the knowledge of the poisons. And it is the science that explains how chemicals, in the very broad sense of the word, that can be chemicals from the outside, that can be chemicals that you form yourself. A hormone that you form is a chemical. How chemicals affect the function of, here we go again, living cells, tissue, organs, organisms. The chemicals can be endogenous, hormones, neurotransmitters, authocoids, we'll see example, or exogenous, natural product, very important in this part of the world, or synthetic. The drugs provided by the pharmaceutical industry. And the chemicals can have beneficial effects or deleterious effects. So every drug that you introduce in the body, well, it can do nothing, but that's very rare. Or it can be relatively good, or it can be relatively bad. And the pharmacologist does measure the production, response to the production of, and the response to chemicals. What happens here? I lost my... What? What should I do?
Is it connected?
It was connected. Okay, let me try. Well, let me continue. You'll see the slides later. The pharmacologist sees what chemicals do, measures how they are produced, if they are endogenous. The hell didn't it work perfectly? And of course, just as physiology needed other science for pharmacology, we need to know physiology, we need to know chemistry, we need to know biochemistry, molecular biology, genetics, etc. Now, a very annoying, and it's too soon to quit, so. Okay, you'll see the slides later, so you have to listen to me right now and not look at the illustration. The very important thing is that when you look at the chemical that you introduce into a body, Paracelsus in the 15th century said, I'll read it because you cannot see it, all things are poisons, and there is nothing that is harmless. The dose alone decides that something is no poison. You can kill with too much oxygen, you can kill with too much water. Okay? Those are the prototypical examples. It's all a matter of dose. And that is why, if we want to define a pharmacologist, and I'm quoting here, you'll see that when you have access to the slide, one of the founders of modern pharmacology, Sir Henry Gaddam. A pharmacologist is a physiologist who knows the right dose, who uses the right dose. And you always have to assume that if you use, as we do on the 4th floor, if you use a chemical to affect the cellular activity, you always have to look into the possibility that you may look at some poisonous effect rather than a physiological function. And I can only recommend to all of you, if some of you ever work with cell cultures and all of that, to remember that, to make sure that you use the proper dose. Now, I think we have to realize how important this is, because every chemical that we bring into our body can affect its function. One of the most frightening article I ever read is a few years ago, when I saw this study that said that in the West, probably the same in Korea, every newborn baby has already more than 200 synthetic chemical molecules in his or her body. So we are slowly but surely poisoning ourselves with too many chemical substances. And that and pollution, to me, are the two biggest dangers for our species. The fact that even in our young kids, youngest children, they already are invaded by chemicals that were not meant to be there. Okay. But as a pharmacologist, it's little I can do there. Now, this must be the thing that blew. What can pharmacological agents do? Well, they can activate the target, agonists at receptors, activators of enzyme. They can inactivate the target, antagonists at receptors, inhibitors at enzyme. They can modify the structure of the protein so that the interaction between that protein and its natural target becomes different. We call that modulators. So they increase the activity or they reduce the activity. It can fool the target. If you have an enzyme that takes substance A and you discover that it has a greater affinity for another substance, by giving that other substance, you can prevent the activity of the enzyme. We will see examples of that. And of course, you also can modify cellular permeability. And I've already given you an example of that with the calcium ionophore. This is basically what we can do. And we can do that at the level of the ligand gated channels, all those molecules that I've given you, we can do that, and you will see those again when you look at I was shown here 3 slides that I already have shown you. And the last part that I wanted to say something about related to genetic manipulation, whereby we now can get into the genome and modify the genetic material that we have. And do you know about that, genetic manipulation? Have you heard about that? Where you prepare an animal which has one protein that is changed, absent, or overexpressed, and that way the whole animal, transgenic animal. Have you heard about that in the back there? Okay. Well, of course, if we suspect as a pharmacologist the importance of a given enzyme, we can knock it out or overexpress it and then look what happens. And again, we have done that, we do that, and I had examples here that we will see in one of the next lectures, but they're in the slideshow if you want to have a look at it. Those genetic manipulations. And the genetic manipulation in recent years has become extraordinarily forceful because one has discovered in the human genome a number of proteins, G protein coupled receptors, membrane proteins that are coupled to G proteins that could be receptors, of which the function is not known. And that provides pharmacology and the pharmaceutical industry with a number of novel targets to look at. But the word of caution that I wanted to bring here is that we should never forget that when you go fishing in the genome, if I take your genome and I look at all the proteins there, certain of the proteins in the genome that are no longer expressed in your cells really are not useful now. They were very important when you were growing in your mother's womb, because this single cell, as it differentiated, we went through different stages. You know, we almost went through a mini evolution in our mother's womb, from one cellular to multicellular to fish-like. We gave up the gills and we started to breathe, etc. Well, there are a number of proteins that are absolutely crucial at those moments. But afterwards, we don't need them anymore. So, genetic fishing is still a lottery as far as I'm concerned. And the example that I was showing here is the example of endothelin, and I will try to remember to come back to that when we discuss it. And from all of that, by altering the activity, my last real slide was to come back to my small unicellular thing, fishing, swimming in the water, and say that through those changes in activity, in all the individual organs and tissues, we can achieve the same goal as what was achieved by the unicellular organism, and we can achieve what we call homeostasis. But as I will stop here because I need my material, and we will pick up from here when I come back in a few weeks, 2 or 3 weeks. Okay? Thank you very much. And next time, let's try to have some backup for the computer. Good. No questions? You understood it all? You're a brilliant class. I'm very proud of you. I'm very proud. Good. I have it all. See you. Thank you. Thank you for your attention.